Scientists at MIT just found a way to turn laser chaos into a brain imaging tool. The discovery began when the laser did the opposite of what people expected. The team was working with a multimode optical fiber, a glass fiber that can carry many patterns of light at once. Normally, when you push more laser power into this kind of fiber, the light becomes messier. Tiny imperfections inside the glass scatter the beam. The output gets less controlled, not more controlled. So the expectation was simple: raise the power, get more chaos. But as MIT graduate student Hong Hao Chow pushed the fiber close to its damage limit, the light suddenly collapsed into a single, thin beam. Instead of spreading into disorder, it organized itself into what the researchers call a pencil beam. That matters for microscopy. A pencil beam is narrow, stable, and extended through depth. Many imaging systems face a trade-off. High resolution usually means a thin slice is in focus. More depth often means more blur. This beam seems to bend that trade-off. According to MIT, the recipe had two precise requirements. First, the laser had to enter the fiber straight on, at essentially 0 degrees. Second, the power had to be high enough that the light started interacting non-linearly with the glass. At that point, disorder and self-focusing balanced each other. The result was order. In plain English, the chaos did not disappear because the researchers corrected every flaw. It disappeared because the system found a stable state on its own. That is the physics story. The biology story is why people outside optics should care. The researchers applied this self-organized beam to multiphoton microscopy, a technique used to image biological samples in 3 dimensions. Their key demonstration focused on a human blood-brain barrier model. The blood-brain barrier is a protective layer of cells around the brain's blood vessels. It helps keep toxins out, but it also blocks many medicines. That is a huge problem for diseases like Alzheimer's and ALS, because a drug cannot help the brain if it cannot reach the right cells. To test brain-targeted therapies, scientists need to see how molecules move through that barrier, whether a drug enters, where it goes, how fast cells absorb it, and whether cell types behave differently. The usual optical approach is slow because it often captures one two-dimensional section at a time. To build a full three-dimensional picture, the microscope has to scan slice after slice. MIT says the new pencil beam captured 3D images of the human blood-brain barrier model about 25 times faster than the gold standard method, while maintaining comparable resolution. That does not mean every brain scan in medicine is suddenly 25 times faster. This is a lab microscopy result, not a hospital MRI upgrade. The careful version is this. For volumetric imaging in the tested model, the pencil beam made 3D imaging dramatically faster, and speed changes the experiment. If a scan takes too long, you only get snapshots. If the scan is fast enough, you can watch biology unfold in time. In the MIT demonstration, the researchers tracked transfer and uptake in a live human blood-brain barrier model, revealing how cells move molecules across the barrier. That is why this could matter for drug development. A team could use human-based tissue models to test whether a candidate therapy crosses the barrier, compare uptake across cell types, and see transport dynamics minute by minute. There is also a practical engineering angle. The researchers say the beam can be produced in a standard multimode fiber with a relatively normal optical setup. But there are limits. This is not a cure for Alzheimer's. It is not a new therapy. It is not a device already sitting in hospitals. It is a Nature Methods paper showing a powerful imaging approach with future work still needed. Still, the idea is beautiful. A beam that should have become more chaotic instead becomes more organized. A nuisance in optical fiber becomes a tool. A physics surprise becomes a way to watch molecules enter a model of the brain's most important gate. The lesson is not that chaos is magic. The lesson is that complex systems sometimes contain useful states hidden until someone pushes them into the right regime. For brain research, that could mean faster screening of therapies that need to cross the blood-brain barrier. For imaging, it could mean a simpler path to high-speed, high-resolution 3D microscopy. And for science, it is a reminder that useful discoveries do not always begin with a perfect plan. Sometimes they begin with a laser that refuses to behave. Subscribe to Science in 5 Minutes if you want clear stories about breakthroughs that are easy to miss when they first appear.